Thank you, guys. I appreciate the invitation to OIS. Very excited to present on Revopsis Therapeutics, which harbors a new era of multi-specific biologics to retina. So our lead asset is RO104. It binds VEGF-A, Ang2, and VEGF-C as a monotherapy. It promises to be a potential for first-in-class, best-in-class asset for neovascular macular degeneration, where 70% of patients have minimally classic and occult lesions. The asset is highly de-risked in that all targets are validated. Regulatory path has the optionality of pursuing a superiority or non-inferiority trial design as it is a monotherapy. And the proprietary format has IP around it through 2044, as all of the CDRs are novel and have been created in-house. Revopsis goes beyond RO104, the lead asset. We have a tri-specific for diabetic macular edema with a novel mechanism of action and a bispecific for geographic atrophy. As RO104 enters the clinic in their phase 1 trial, these two assets will be in IND-enabling studies. So the field itself has revolved around marginal improvements in durability since the approval of Lucentis. Every single new therapy that's been approved has been approved through a non-inferiority trial design. And why is this? This is because every therapy has been targeted against VEGF-A alone, with Vabysmo being the first bispecific. We know that there are multiple pathways that can lead to angiogenesis. VEGF-C and Ang2 both are upregulated when you have inhibition of VEGF-A. So what happens if you inhibit multiple targets at once? From our friends at Opthea, we see with minimally classic and occult lesions specifically 16.1 letters of gain in from their phase 2b trial, which compares favorably to every other market authorized drug to date in terms of their pivotal trial. RO104 promises to combine Vabysmo, the best aspects of, with that of OPT-302 in a single agent monotherapy. It promises to provide superior vision and sustained vision for our patients. There's a low risk for immunogenicity as the sequences itself are fully human, and it lacks an FC, which reduces the immunogenicity of the compound. It has high matched picomolar binding affinities, and it's been proven to be scalable and manufacturable as we already have it in cell lines and are creating drug substance. So RO104 has been significantly de-risked to enable an IND in 2025. The targets, again, are validated. The regulatory path is clear. We have completed a Type B meeting with the FDA. And as a monotherapy, we have the optionality in terms of our trial design. The pharmacology and pharmacokinetics have been established. The risk of immunogenicity is low, and the manufacturing process and CMC is already underway with drug substance being produced. Toxicology studies will be started in January of 2025, and most importantly, we plan to be in the clinic next year with a top-line data readout from a phase 1/2A trial at the end of 2026. We've also completed NHP laser CNVM model, which was overwhelmingly positive. RO104, most importantly, was well tolerated at both the high and low dose groups, with no incidence of intraocular inflammation in the high dose group. Impressively, RO104 was able to cause complete regression of grade 3 and grade 4 lesions in this model, and essentially a 2.5-step reduction in the most severe lesions, grade 4 lesions. This data compares favorably to that of faricimab and aflibercept in comparable historic studies. The leadership team is made up of a team of experts from bench to bedside, with Ramesh Bhatt as our CSO. And the advisory team consists of some of the luminaries in the field, with Mark Barakat, Jeff Heyer, Peter Kaiser, and Arshad Kanani advising on the phase 1/2 clinical trial design. And most recently, Emmett Cunningham coming on board as one of our strategic advisors. Thank you so much. We appreciate your time today. And I look forward to actually presenting the update next year at this conference.